Hello.
Hello.
Hi, how are you doing?
Good. Thank you, Professor Lin, for giving your speech today. We are very honored to have you. So I'll introduce our speaker. I'm really honored to introduce our keynote speaker today, Professor Xihong Lin, who will talk about a multi-dimensional integrative scoring framework for predicting functional variants in the human genome. Professor Xihong Lin needs no introduction, and I think that covering all her achievements will be a long talk in itself. So I will try to cover some key points here quickly. Professor Lin is professor and former chair of the Department of Biostatistics, coordinating director of the program in quantitative genomics at the Harvard T.H. Chan School of Public Health, and professor of the Department of Statistics at the Faculty of Arts and Sciences at Harvard University, and associate member of the Broad Institute of Harvard and MIT. Dr. Lin's research interests lie in the development and application of scalable statistical and machine learning methods for the analysis of massive data from the genome, exposome, and phenome, including big and complex genetic and genomic epidemiological and health data. Dr. Lin has been supported by numerous prestigious grants and awards, including R01 and PO1 grants from several federal agencies, including the NCI and several other granting institutions. Dr. Lin was active in the early phase of the COVID-19 pandemic. She's a corresponding author of the JAMA and Nature papers on the analysis of the Wuhan COVID-19 data on transmission, public health intervention, and epidemiological characteristics. She is the senior author of the 2021 JASA discussion paper on modeling COVID transmission dynamics in US. In spring 2020, Dr. Lin served on the State of Massachusetts COVID-19 Task Force and testified in the UK Parliament's Committee of Science and Technology on COVID responses. Dr. Lin is an elected member of the National Academy of Medicine. In fact, she has been recently elected as a member of the National Academy of Sciences in 2023. She received the 2002 Mortimer Spiegelman Award from the American Public Health Association, the 2006 Committee of Presidents of Statistical Societies, that is the COPS President's Award, the 2017 COPS F. N. David Award, the 2008 Janelle L. Norwood Award for Outstanding Achievement of a Woman in Statistics, the 2022 National Institute of Statistical Sciences Jerome Sachs Award for Outstanding Cross-Disciplinary Research, and the 2022 Marvin Zelen Leadership Award. She is an elected fellow of the American Statistical Institution, the Institute of Mathematical Statistics, and the International Statistical Institute. Dr. Lin is the former chair of the Committee of Presidents of Statistical Societies, that is COPS, and a former member of the Committee of Applied and Theoretical Statistics of the National Academy of Science. She is the founding chair of the US Biostatistics Department Chair Group and the founding co-chair of the Young Researcher Workshop of East North American Region, that is INAR, of International Biometric Society. She co-launched the section of Statistical Genetics and Genomics of the ASA and served as a former section chair. She's the former coordinating editor of biometrics and the founding co-editor of statistics in biosciences. She has served on a large number of committees of many statistical societies and numerous NIH and NSF review panel. Professor Lin, I'm very honored to introduce you, and it's over to you.
Thank you so much, Samisa. This is a very generous introduction. And also I want to thank the committee for inviting me, and very honored to give a presentation as the NIST students event. So today's talk I will be focused talk on a particular topic that was the committee asked me to speak on. So let me share the full screen. The full screen now?
Yes.
Okay, excellent. Thank you. And so I was asked to talk about the how to predict variance function and in the whole genome. And so therefore this talk will focus on that aspect. And so this work was co-led by my former students, Xihong Lin and Godwin Yang, and also my computational biologist, Hufeng Zhou, and in collaboration with Juliana Anita Laza at Columbia. And so this paper was published in American Journal of Human Genetics last year. So let me first talk about the genetic variance functionality, and so why we care and why that is useful. And then, so let me start from the whole genome sequencing timeline. And so about 15 years ago, that was the completion of the 1,000 genome. At that time, the sequencing cost was pretty high, so therefore the depth was pretty low, and only a couple thousand people were sequenced. And in the last couple years, and the sequencing cost has dropped dramatically, and several large whole genome sequencing consortia has been launched in NIH. One is called TopMed. This is called the Transomic Precision Medicine Program by NIH LBI. So it has sequenced about 190,000 samples. The other is called the Genome Sequencing Program by NIGRI. So that has sequenced about 140,000 whole genome, over 200,000 whole exome. And also NCI has its own sequencing effort. So they will sequence about 200,000 samples, and including tumor. And so in the last couple of years, several large-scale biobank has been launched globally. And the most well-known one is the UK biobank. It has sequenced about 500,000 whole exome, and also right now about 200 million whole genome. And by the end of the year, there will be 500,000 whole genome sample available. And then the data are integrated with electronic medical record and epidemiological data. So Million Merchant Program, it will sequence about 100,000 samples to start with. It has enrolled about 1 million people. So basically, we are going to have a lots of whole genome sequencing data. So give you a sense how big the data are, and this is the type of data we work with. And so in short, the data we work with are very large. And so this is the sample to show you how big the data are. And so in TopMed phase 10, it has sequenced about 185,000 whole genome. And before the QC, there's about 1 billion variants. So therefore, you can think about the data as 185,000 rows at 1 billion columns. And so with the very large data, and so generally majority of the variants are very rare variants. And so therefore, in order to analyze those rare variants after the QC, you can see about 845 million variants are observed. And then about 45% are singletons, 16% doubletons, only greater than less than 1% are common variants. And so therefore, in order to analyze those massive whole genome sequencing data, and especially those rare variants and the functional annotation are critically important. And so that is how we are interested in the functional annotation. And so why they are useful. So they can help prioritize the top GWAS or whole genome sequencing trait associated variants for experimental follow-up. They can also help with the functional fan mapping. They can also help with heritability estimation, and also they can help with the real variants whole genome sequencing study. And so therefore, the functional annotation of the whole genome data is a critical important first step. And so not everybody is familiar with genetics. Those will be a very quick introduction of the gene of the coding and non-coding variants. And so human genome has 3 billion positions. And so basically the letter ACTGs. And so you can think about those letters across the genome. And so for each positions, and generally there's one reference allele and the 3 alternate allele, because there are 4 letters. So therefore, if there's one reference allele, then there are 3 alternate alleles. So therefore, totally there are 9 billion variants. So there are totally about 220,000 genes. So the genome is composed of the gene and also intergenic region. The gene is coded by purple, and the intergenic region is coded by yellow. And within each gene, and that is can be decomposed into the exome, and which is coded by red, and also intron, and this is coded by green. And within each window block, and then basically there are lots of variants. And so the coding variants, they are mainly located in exon, and basically those white part within the gene. And the non-coding variants, and they are located in the yellow part and the intergenic regions, and also in the introns. And so think about this across the genome, there are total 3 billion positions and 9 billion variants. And so we need to functionally annotate every single position in order to facilitate analysis. So what do we mean by functional annotation? So when we talk about variants, and then there is the diverse set of functional annotation. And so this can be captured into following categories, including the protein function that are measured and for the coding variants, and were predicted for the coding variants, and also evolutionary function that basically measures the conservation or the epigenetic or biomedical evidence, and especially for the non-coding regulatory variants. And so then we want to see how the different variants affect the disease outcome, and then which variants are likely to be functional, then they are more likely to be the drug target. And so then what we mean by the protein function? Protein functions are only relevant to the coding variants for the exome. So if you look at here, so for example, look at this particular location. And so this, so you can see that, so basically a set of DNA sequence will code amino acid. For example, CAT will code this amino acid HIS. If this particular variant A, suppose there is a mutation, and then change to C, then you can see that will code a different protein and different amino acid, and that will produce a malfunctioned protein that could cause the diseases. And so therefore protein function is critically important. We want to see whether a particular variant has a particular protein function. And so what is evolutionary conservation? So basically, so if you look at this part on the left, so if you look at here, you can see for this sequence, it is preserved across the different primate, human, chimp, and bonobo, and mouse. And so these variants here, they are quite conserved across different species. If you look at the figure on the right, then you can see for this sequence, and so you can see the sequence vary for this particular region, the sequence vary from the human to chimp and to mouse. And so this region is not that conserved. So generally for the conserved regions, and they are more likely to be functional. And so the last part is regulatory epigenetic function. This is more relevant, and for the non-coding regions, especially for intergenic region. So basically, one want to see whether a particular variants and that can play a regulatory role to regulate the gene expression. And so therefore, many, especially for many GWAS findings, and they are mainly in the non-coding regions, and so they are mainly playing the regulatory roles. And so therefore, so the regulatory region generally include the promoters, that is the upstream of the gene, and or enhancers that can be further away from the gene. And so if one look at the non-coding variants, as I mentioned, that non-coding variant consists the intergenic region, that is that yellow region, and also the introns, meaning the intergenic region, that yellow region. And so in order to measure the functionality of those variants, and so generally the ENCODE consortia play a critical role. And it generates many epigenetic score like a K27ACs and ME1 and ME2. And so also one can also estimate the conservation scores. And so suppose here there are about 20-ish epigenetic score from ENCODE, and then there are 10-ish conservation score. And but if you look at the data here, you can see this heat map among the epigenetic score, there are a good correlation among the epigenetic score of those variants. But among the conservation score, you can see there are also a good amount of conservation correlation. However, there's not much correlation between epigenetic score and the conservation score. So what this tells us is when we talk about functionality of a particular variant, we cannot measure the functionality using a categorical variable, yes, no, whether the variance is functional or not. So we need to use multidimensional measure. So for example, whether the variance has regulatory function, yes, no, or the variance has a conservation, and yes, no. And so this tells us basically it's useful to use multifaceted annotation. So now how can we do that? Because if you look at the ENCODE score, and there are about 20-ish ENCODE scores, and then here's about 10-ish conservation score. So how can we construct a particular location, whether this variance has a function, epigenetic function, or conservation that is more like a binary? And so this is not observed. So basically those are the observed data. And so we want to use this observed data to infer whether a particular variance in a genome is functional or not, in what dimension. So that is come up with this integrative score framework. So from that heat map, it illustrates that it's not desirable to simply classify variance as functional, yes or no, just a binary, because when we talk about functionality, it needs to be measured in multi-dimension. And so the variance functions are multifaceted, and including the protein functions, conservation, regulatory function. This cannot be measured using a single integrative score. And so therefore, this suggests that we want to create multidimensional integrative measurements of functionality. And so how can we do that using the observed data? So multiple effort has been made and to construct the integrative functional score. And so they include the supervised method, and for the supervised method that are based on the labeled training data. And for example, the popular methods such as CAD or FTHMM, and they are constructed using the support vector machine and based on the labels. And also the DAN, this is also a supervised method, and that is based on the deep neural network and also based on the labeled data. And also this GABA, this is based on random forest, is again based on the supervised method. However, the selected training data set generally is biased. And so for example, one use the clean variants and to do the training, but the clean variants generally are quite selective. And so they are meaning the loss functional variance or missense variance or the very conserved variance. And so it doesn't capture the genome very well. It's basically the bias sample of the variance across the genome. So that is a limitation of the supervised method. And several efforts has also been made to propose unsupervised method, which do not depend on the labels. And so this includes Genome Canyon and Eigen, and also the PAN and the FAN LDA. So, and also there are several semi-supervised methods. And so the so I'll show you what the limitation of those supervised unsupervised method. For example, Genome Canyon, so they only use unsupervised method and across the genome. So therefore, the samples of the variants are not that biased because they use all the variants across the genome to build the unsupervised model. But the problem is it creates only a binary indicator, and it will indicate if a variance is functional or not. So as we discussed, it's not desirable to create only a single binary indicator to indicate variance is functional or not, because the variance generally have multifaceted function. And so there are also several semi-supervised methods, and such as the Lin side. And those are evolution-based method. And so they work well for the coding region, they work well for the promoters, and because because it rely on protein function and also scores, and also the conservation scores. And generally they work well for the promoters, but they don't work well for the enhancer, and because many of the variants in the enhancer regions, and they are not conserved. And so this method, those integrated score method, generally take either the continuous scale or probability scale. And so the advantage using the probability scale, it has a natural interpretation that indicates the posterior probability of whether the variance is functional or not. And like a cat and eigen, they are continuous score based integrative score. And then like a Dan and the Lin side and Genome Canyon, they are all posterior probability based score. And so what are the major limitations? The major limitation of all those methods is they assume a single integrative score. And so that indicate if for a particular location, if the score is high, that indicate the variance is functional. If the score is low, that indicate the variance is not functional. And as we discussed, this is not desirable because the functionality of the variance are really multifaceted. And so this basically motivate this, we call the MACIE. This is the multidimensional annotation class integrative estimation. So what is a MACIE? What does MACIE want to do? So the goal of MACIE is to develop a multi-dimensional probability score and to characterize multifaceted of the variance function using unsupervised learning framework. And so how can we do that? So basically the idea is to integrate a large number of individual functional annotation, like a protein function and conservation and epigenetics function, and then create multiple latent functional class. So here are the original data. And so the data basically here, this example indicate the available conservation score. So you can see there are multiple, about 10 conservation scores. So, but we don't know whether a variance is conserved or not. So functional status is not known. And so basically this is a missing data problem. We have all those measured features, but we want to infer using those measured features to infer whether the variance is functional in what dimension, and whether this variance is conserved, or this variance is has a regulatory function, or this variance has a protein function. And so if you the existing methods such as Genome Canyon, it uses a single latent variable method, single latent variable framework. So basically the latent variable here is just CI, it's a scalar, and to indicate whether a particular variance is functional or not. And then conditional on this unobserved latent class, and so this indicates whether variance is functional or not. Then the observations of those scores, such as the conservation scores and also proteins functional score, they are assumed to be independent. And so here you can see there are multiple limitations. First is assume for each location, and the variance is functionality is measured by a single latent class, whether the variance is functional, yes or no. And also conditional, but we know, like from the example I showed, that we cannot talk about functionality of a variance in a single dimension, whether the variance is functional or not. It can be functional in epigenetic function, but may not be functional in conservation. And so therefore, we don't want to create a single latent variable. A second limitation is this conditional independence assumption. So that means conditional on the latent variable, latent class, and then all those continuous scores or categorical scores are assumed to be independent. And this is a strong assumption as well. So therefore, MACIE try to do is assume multiple latent classes. And so for example, the first latent class is whether the variance is conserved or not, yes, no. The second latent class, if the variance has a regulatory function, yes, no. And so in this situation, then you can see there are two latent classes. And then those two latent classes follow unobserved, they are unobserved. And then they are assumed to follow a multinomial distribution. Then, so this is the first feature. So allow multiple latent dichotomous functional class, for example, conserved and regulatory function, yes, no. And the second feature is conditional on the latent class, relax the conditional independence assumption, and allow those observed scores, observed feature to be correlated, and within each latent class. And so therefore, the model will be more robust. And so then the Ys here are observed, but the latent classes are unobserved. Those are missing data. So this basically can be set up into this latent mixed effect model framework. Suppose we have latent class multivariate binary vector. And suppose like the status is a vector C, and it takes a value CI1 to CIM. M indicates the number of the latent classes. And so for example, suppose M equal to 2, then for the non-coding variance, and we have an indicator to indicate whether variance is conserved or not, yes, no. And for C1, for C2, that will indicate whether the variance, the same variance has a regulatory function, yes, no. And so in that situation, M equal to 2. And so in this situation, you can see the C will take a combination, totally the C vector will take 4 values, 1, 1, 1, 0, 0, 1, 0, 0. So that's why the multinomial distribution will be reasonable. And then, so therefore, this latent variable, CI will follow a multinomial distribution. And with this gamma will indicate multinomial probability. And the CI is not observed. And so here, to set this up, I will indicate the variance. And so from the training data, we can have the N basically will be 1 million. And then after we build the model, we apply this to the whole genome to predict the function across all 9 billion variants. And the J basically indicates the latent classes. J goes from 1 to M. Suppose for the non-coding variants, we'll make M equal to 2. Basically, we have 2 latent classes and variable. One to indicate conservation, yes, no. The other to indicate regulatory function, yes, no. And this K basically indicates the functional score in a latent class and take value from 1 to Lj. And for example, for the conservation, this L will be 10 conservation score. And for epigenetic function, this L will be about 20 epigenetic score from ENCODE. So what is the model? So conditional on this unobserved latent classes and the Cij, and then we allow the observed annotation scores are to be continuous or discrete and follow a factored generalized linear model. And so here, then we allow different link function because some of the annotation score are continuous, some of the annotation score are binary. And for continuous annotation score, we can use a linear link. For the categorical annotation scores, we can use the logit link. And then this basically assume the observed annotation score related to this latent class variable and through a generalized linear model. And so it can allow the linear link, or if it's a count variable, can use the log link. And this BI basically is the random effect. So this random effect is assumed to follow a normal distribution with mean 0 and also this factor analysis covariance. And so in other words, if we go back here, and so basically random effect model the correlations, conditional on the latent class, random effect model the correlation among the observed scores. And then because we assume the latent variable, the two latent variable, like this is a conservation class, and this is a regulatory function class. And because they assume this follows a multinomial distribution. So therefore, if you integrate out the C's and then the Y's, all the Y will be correlated. So we account for the correlation and through this multinomial distribution of the C and also the joint distribution of the Y conditional on C. Okay, and so then this become a missing data problem. And the observed data basically observe the annotation score, such as this 10 constellation score and then 20 epigenetic score, and also a 10 protein functional score. And so unobserved data are the latent class, CI, and also the random effect. The parameter will involve the regression coefficient and also the factor analysis covariance in the distribution of the random effect, and this gamma is the probability parameters in the multinomial distribution of the C. And so what is the goal? The goal is to fit this model. This is generalized latent generalized linear mixed model. And then after we fit that model, we estimate the parameters, and then we calculate the posterior probabilities and of the latent class and giving the observed data. Then this will tell us the latent functional class probabilities. And then this will provide information to indicate whether the variance is conserved and whether the variance has a regulatory function and also what's the probabilities. And so to implement this, because the observed data are the Y's, the missing data are the C and the B's. And so this can be implemented using the EM algorithm. And so this first part is a conditional likelihood of Y giving the latent class and the random effect. And so the second part is the likelihood for the random effect. The third part is the likelihood, multinomial likelihood of the latent class. And now, then this can be done through the EM. And so the E step, basically you calculate the conditional expectation of the joint likelihood, conditional on observed data. And then the E step, after you do the expectation step, and then you can do the M step to maximize this conditional likelihood function. Then by doing that, you can also calculate the byproduct of this posterior probability of the latent class giving the observed score. So you iterate this until convergence. And so the goal is to estimate this probability of a C giving Y. This basically measures the functionality of the variance giving the observed score. So in order to do this, and so this you can see there's no label at all. So this is completely unsupervised. And so we need to show and this works. And so especially in genetics, it's very important. You can develop many different fancy methods, but I had to really show that really work. And then in order for people to feel comfortable using the method. So therefore, we trained the data using multiple data set, and also we benchmark the result using several data sets as well. So when we train that model, and we use the non-synonymous coding variance, and also to train the model to construct the functional status, a class status for the coding variants. And for non-coding variants, and so we basically use a non-synonymous coding variants and the non-coding variants, and to train the model to construct the posterior probability of the functionality in terms of conservation and the regulatory function, giving the scores. For both models, we took 2 latent classes and m equal to 2 to help with the model interpretation and computational time. And for the coding variants, and so the 2 classes basically indicate the protein function that has and the conservation. And then the Y for the protein function has a dimension 4. So that means we have 4 protein functional score. And for conservation, we have 8 conservation scores. The C's are not observed. So we totally use the 2.2 million variant to train the model. And for non-coding variants, we also consider the 2 functional class, including the conservation and the yes/no, regulatory function, yes, no. And so therefore they are also these 2 C variables, and but the 2 C variable interpretation for coding variants and for non-coding variants, they are different. For coding variants, and those 2 latent classes are the protein function, yes, no, conservation, yes, no. For non-coding variants, the 2 classes are conservation, yes, no, and the regulatory function, yes, no. And so what are the Y's? The observed score, we have 8 conservation scores, and also we have 28 epigenetic scores and from ENCODE, including 3 histone marks, 12 open chromatin marks, and 3 transcription factor scores, and the distance score, and also the chromatin state variable, and the GC, and the others. And totally we trained using about 40,000 variants. And then we fit this generalized factored generalized linear mix models, and then by fitting those models, and we can calculate the posterior probabilities, and using the EM algorithm. And now we need to show this really works. So in order to show this really works, and we did a lot of benchmarking using many different data sets. So the benchmarking in genetics is very, very important. And so I had to really show they really work from many different data sets, and especially if you want to publish in top genetic journals. Okay, so this is the first testing data set for this is the coding variants, first testing data set. So we use the ClinVar non-synonymous coding variants as a testing data set. So in this situation, the true labels are the pathogenic benign. Pathogenic variance is coded 1, and the benign variance coded as 0. So this is the true label from the ClinVar database. And so we use the MACIE to predict the labels, and without looking at the true label, and then construct the ROC curve to see how good the prediction is. So by looking at the ClinVar variants, as I mentioned, they are mainly the coding variants, and so including the loss function variants and missense variants, and also they are many of them are conserved variants when the ClinVar variants are selected. So ClinVar database is not an unbiased database; it's a biased database. They only select set of variants which are of clinical interest, and so therefore many variants in the ClinVar database are conserved variants, and also loss functional variants or missense variants. And so this is not unbiased sample. So therefore, if one use ClinVar to train, and then this training data set will be biased. But when we use unsupervised learning and to build a prediction model, we use all the data, and so therefore the training data set will not be biased. So now if you look at the figures on the left for the non-synonymous variants, and so you can see the MACIE protein function, ROC, this is a blue curve, and the AUC is about 93%, and conserved, and this is this kind of the gray curve, this is about 89%. And so because the ClinVar variants are mainly selected by loss functional variants, so they have high protein function, so that's not surprising. So protein functional score have a better prediction. And now this yellow basically indicate any class, either the protein functional class or conserved class. So basically protein functional class, this is a binary latent variable, take value 1, 0. Conserved latent class is also binary 1, 0 as well. So you can see this outperform the other methods such as FATHM and also the CAD and eigen as well. If you look at the figure on the right, and similarly, and this blue indicate the AUC. But here I want to say the CAD work quite well, and for the coding variance, and that makes sense because in the training data, CAD use the code based on the evolution, and then on the conservation. So that's why CAD work quite well here. And if you look at the missense variance, you can see the MACIE also seem like this dark blue curve, and also seem like perform very well. And also it outperformed the CAD and eigen and so on. Has similar performance at eigen, and but outperformed the others such as DAN and the FICON. So then this is the second coding test data set and to validate as a benchmark. So in this data set, we use the loss functional variance in BRC1 and for this SGE data set. And so again, so here the loss functional variance versus functional variance in the BRC1. This is a true label, and then we use a MACIE to predict this true label. And so again, you can see this purple based on MACIE protein function has AUC 91%, and MACIE based on conservation 81%, MACIE any class 88%. This outperform all the others. If one look at the BRC1 loss functional variance versus the ClinVar benign variance, you can see this MACIE also work well is compared to the other method as well. So then we benchmark looking at the non-coding variance prediction performance. So in this first benchmarking, so they use the ClinVar non-coding variance and using pathogenic non-coding variance as 1 and benign as 0. So this is a true label. So if you look at here, you can see that protein regulatory MACIE regulatory variance here, it doesn't have much a good performance here because the pathogenic variants are mainly conserved. So if you look at the MACIE conserved, and you can see the ROC work very well, it's 95%. And so this has very similar to Linside and CAD. And so that makes sense because many of the variants here are conserved variants. So therefore MACIE conserved work well, this class work well. And so now if you look at next benchmarking, so here we use the cage promoter and the enhancer of 1000 genome data, and they use the cage to define the promoter and enhancer and as the regulatory function equal to 1, and those variants not in the cage define the promoter enhancer as 0. So if you look at the here cage promoter variance, you can see the purple, that is a MACIE regulatory variance, AUC about 75%. And for any class, the MACIE conserved is the AUC about 63%. For non-coding variants, the two classes are regulatory function and conserved. So because for promoters, and many of the promoter variants are conserved, so therefore you can see CAD still work quite well, and the Linside also work quite well. However, if one move to the enhancers, and then you can see that MACIE AUC about 78%, and the conserved MACIE is AUC about 62% using any class, either regulatory or conserved, that is about 75%. So that, but if you compare that with CAD, the AUC is only 0.6% or eigen 0.7%. And so therefore this indicates that the CAD does not work well for the enhancer, but CAD work well for the promoter, and MACIE is robust for both the coding and non-coding variants. And also we'll finally look at this last testing data set and using the MPRA. This is this data set. And so you can see for this data set using the reporter assay, and the MACIE still outperform all the other methods. So for example, MACIE regulatory AUC is 68%, but CAD is only 57%. And then we apply this to the GWAS summary statistics. So because many of the GWAS variants may not be functional, so we want to apply the annotation to see which variants are likely to be functional. And so here is LDL. So for this LDL, this is the bad cholesterol, and so the R7412, and this is a coding variant. So if you look at R7412 and this variant, when we predict using the MACIE protein score and the MACIE conserved score, you can see those are the posterior probability, then they are very close to 1. So that indicates this variant is highly functional in both in terms of protein function and also in terms of a conservation. And if you look at this variance nearby, and then the predict, even this variance is highly significant in GWAS analysis, has a highly associated with LDL. But if you look at the MACIE conserved score, and then that is less than 1%, that means this variance is not very conserved. And so this MACIE regulatory variance score is 99%. So that means this variance has a high regulatory function, but not much conservation. And also we apply this to HDL as well for this region. And so for this variant, you can see these variants are all very significantly associated with HDL. And but if you look at this particular variant, RS 17, 23, 1506, and so even this variant has very, very small p-value associated with HDL. So but doesn't have much function in terms of conservation and regulatory function. If you look at these two variants, even they are not as significant as the first variants in terms of the GWAS p-value, but you can see this variance here. This has a conserved score less than 1%, but has a regulatory score probability 99%. So that means these two variants have a high regulatory function. So therefore, when you do GWAS analysis, the most significant GWAS SNP may not be most functional. And so you want to look at the nearby variants to see which one may not be as significant as the most significant one, but they are more likely to be functional. And so to summarize, so MACIE is the unsupervised learning framework that integrates multiple functional annotation. And so MACIE output integrated multidimensional, easy to interpret probability, posterior probability score to capture multifaceted variance functionality. And also it flexibly combines multiple annotation by allowing both continuous and dichotomous annotation, and also multiple latent functional status, and also between and within group correlation. So it also, we use the multiple testing data set to really check whether really MACIE result makes sense. So it shows that it provides a powerful and robust performance compared to the existing integrative functional score method. And also it can help prioritize functional variance using the lipid. And also we apply that to the lung cancer GWAS data as well. And also we use this MACIE model to generate a posterior probability across the genome for all 9 billion variants. And so this can be downloaded from this website. So just to show you application about how the annotation can be useful in the whole genome sequencing study. So for the genome sequencing, NHGRI genome sequencing program, and we have a functional annotation working group that create a functional annotation database we call the FAVOR, and that integrate functional annotation score across many different sources from ENCODE, roadmap, CAD, and GTAC, and so on, and provide annotation of all 9 billion variants from allele frequency, conservation, protein function, epigenetic score, and so on. And so it has over 300 annotations. 80% of them are built on build 38. And so this database was published in the Nucleic Acid Research, a special database issue earlier this year. And so this favor backend database is publicly available, and also one can use this online portal to query the annotation of any variants or any region or any gene. So this work was co-led by my computational biologist Hufeng Zhou and software engineer Ted. And so they took a lead in developing this relational database. I think I never thought in my life I would develop database, but I think that like in order to do the statistical genetic research, while the especially for whole genome sequencing analysis, this database development is critical. And so that's why I hired a software engineer, I hired a computational biologist to help build this relational database, and then that can allow for very fast query. So to see how that works, suppose you have the TopMed data or whole genome sequencing UKB data or GSP data. First, you run the QC and the VCF file, and using Hill, the developed by my colleague at Broad by News Group, that will generate the QC, the genotype file. And then you have this backend database, favor backend database, which is available in the cloud. In the this is NHGRI ecosystem called Anvil, and or NHLBI and the cloud ecosystem called Biodata Catalyst, or the UKB, the whole genome sequencing data cannot be downloaded anymore. So this had to be analyzed in RAP and using the DNA nucleus. So you have this backend database. And then what you do is we develop this favor annotator, which can annotate any whole genome sequencing study. For example, like TopMed has 1 million variants, then one will use this 9 billion variants as a backend database to annotate those 1 billion variants in TopMed. That will create functionally annotated TopMed whole genome sequencing data. And then one can perform the association analysis, such as the real variance association analysis. And then it turns out if you want to annotate the TopMed 1 billion variants is in the cloud, is with 1 billion variants is only take 3 hours and $24. So that's very affordable. So that is good. And then this annotation database is favor annotation database. They are public available, so everybody can download it in Harvard database. And for those annotated whole genome sequencing data, like a TopMed data, they are available in the NHLBI data common, Biodata Catalyst, and also GSP whole genome sequencing annotated data are available in Anvil. This is NIGI data common. For UKB whole genome sequencing data, they are the annotated data. We are going to make them available in RAP. And so I won't have time to go through this, how to incorporate annotation in real variance association study. And so this work was co-led by my former students, Xihong Lin and Xihong Lin. And so basically the idea is to incorporate the functional annotation to dynamically upweight functional variance when we develop real variance association tests. And so this work we call the STAR, and this work was published in Nature Genetics 2 years ago, and also in Nature Method last year. And also for meta-analysis and using the STAR framework, and this was published in Nature Genetics earlier this year. So the last 2 minutes, I want to mention it's important to get involved in the cloud. And so the NIH and also many biobanks have put a lot of effort to create a cloud-based ecosystem. And so this is a trend. And so we need to join the force. So give you an idea of what is the traditional way for data sharing. The traditional way for data sharing basically is the data copy. So for example, for UK Biobank data, the old way is everybody copy the UK Biobank data to our own institutional cluster. And so basically the no sharing of the infrastructure data copy is the same as the data sharing. But with the whole genome sequencing data, this strategy doesn't work anymore because the data are too big and to copy it to individual cluster and also too expensive to store in individual cluster. So therefore, this new biodata ecosystem is cloud-based. So basically bring the researcher to the data. So there are multiple cloud environments such as Google Cloud, Amazon Cloud. And so so one need to learn how to develop the workflow description language and develop the cloud applet. So the examples include the unviews and also include the biodata catalyst and include the DNA nucleus by UK Biobank. So that is important to learn and to work with the cloud environment, and so learn how to do distributed computing, how to do federated learning. So I think I will stop here. We have 5 minutes and for question, comments, discussion.
Thank you, Professor Lin, for this wonderful talk, and you've touched on such incredibly important topics today. I'm sure this is going to be very helpful to us all, and especially ones working in genomics. Do we have any questions for Professor Lin? The floor is open.
I have a question. First of all, thank you so much. That was a really cool talk, and I especially liked how you put it out an open source tool for people to use. I think that's really cool. So I had just like a point of curiosity. When you're doing the ROC curves of the different methods, I think this was like slide 28. There was that one method, the fit cons, that did very poorly. Like sometimes it looked like it had an area under the curve of like less than 0.5, so it's doing like worse than a guess. I was wondering why if you had any thoughts on why that method just did very poorly across the board.
Yeah, I think that's a very good question. So the different method is based on the different set of score, and some method are supervised, some method are unsupervised. And for FitCon, if you look at here, so let me go back. Okay, FitCon here. And so this is based on, oh, where did that go? FitCon here. This is semi-supervised, okay? So that's mainly based on evolution. So evolution scores basically capture the conservation, okay? And so therefore you can see the FitCon will work well for the promoter regions, but for enhancers and FitCon will not work well. So that's why the AUC is low. And because for the enhancers, when we will need to include not only the conservation score, but also epigenetic score as well. So that's why even people say we use a supervised learning or semi-supervised learning, doesn't mean it will work well robustly across the whole genome. It really depends on what training data set you use and also what type of a feature you use. Make sense?
Yeah, that does. Thank you.
Yeah.
We don't have any more questions. We will close this session today. Thank you again, Professor Lin, and thank you all for attending this talk.
Thank you for having me. Enjoy the rest of the meeting.
Thank you.
Bye.
Bye.